Hello everybody, welcome to the MDC webinar series. I'm Sarah Brockwag, I'm the lead scientist at Medicines Discovery Catapult, and I'm hosting the session with my colleague Dan Bond. So we're now on to the fifth in this series of weekly webinars that runs on a Wednesday at 2 p.m. So as I say, we're on to the fifth in this series of 9 informative drug discovery sessions. So each of the sessions is delivered by experts at the Medicines Discovery Catapult and our CRO network partners. And each of the webinars is focusing on an aspect of preclinical drug discovery. We started with selection of the right target and identifying the hit. We've gone through optimizing the compound, which is the subject of last week's webinar, through PK/PD, disease models, and finally defining safety and biomarker strategies. So in this fifth session, we're thinking about chemistry and formulation, and I'd like to welcome 3 speakers. Firstly, Trevor Perrier, Chief Scientific Officer at DomainX, and Trevor's going to talk about fragment-based approaches, taking us through a case study of approaches to turning fragment hits into drug candidates. Then James Hitchen, who's Head of Medicinal Chemistry at Charnwood Molecular, is going to focus on synthetic chemistry aspects of drug discovery and early development projects. And finally, Alison Foster, who's Head of Technical for Preclinical Services at Key Pharma. Alison is going to get us thinking about the design of formulations for early stage preclinical and clinical studies. We'll take questions at the end of each of the presentations, and if you wish to ask a question for the speaker, that is, please type these at any time in the Q&A box at the bottom of your screens, and the speakers will address them at the end of their talk. So over to you, Trevor.
Right, thank you, Sarah. I hope you can all see my screen. Welcome to the webinar, everybody. This afternoon, I'll be presenting the example of fragment-based drug design carried out by the team at DomainX. And I'd like to start by telling you, with one slide, just telling you a little bit about DomainX. So we are located about 10 miles southeast of Cambridge city centre. We're currently 50 to 60 scientists, and we have protein scientists, assay biologists, and medicinal chemists all working together on drug research projects. This is our building here on the Science Research Park. And we're a dedicated CRO. So for the last 3 years, we've had no ongoing internal research projects. All of our work has been supporting client programs, and our clients range from big pharma through to academic institutions, but the majority of our work we do for biotechnology companies around the world, mostly in the UK, USA, Europe and Australia. And as a result of that work, a number of compounds have gone into clinical trials. Other compounds are currently in the preclinical development pipeline. And because of our contributions to those projects, we've been cited as inventors on more than 60 projects. The program I'm describing today was aimed at a group of inflammatory diseases known collectively as interferonopathies. This is a class of autoimmune diseases which are caused by upregulation of interferon signaling, and that leads to inappropriate activation of what's called interferon-stimulated genes, many of which are pro-inflammatory mediators. And major examples of these so-called interferonopathies are systemic lupus erythematosus, Sjögren's syndrome, and scleroderma. Just zooming in on lupus in particular as an example, there are about 500,000 cases of lupus in the G8 countries. And in fact, in China, there are more patients with lupus than with perhaps the better known disease, rheumatoid arthritis. Here is a photograph of a young person who unfortunately has lupus. As you can see, it causes these extremely painful and unsightly skin lesions But even more tragically, it also leads to damage of internal organs, such as the kidney. And because of this, unlike some other immune diseases, it can be fatal, and a significant number of lupus patients, in fact, die of renal failure. Lupus can be treated with steroids and with methotrexate. And in fact, a drug that's very much in the press at the moment, hydroxychloroquine, is also used to treat lupus, but its precise mechanism of action, as per its muted action in COVID-19 treatment, is in fact unknown. In common with other immune diseases, there is a biologic treatment, a BAFF inhibitor called belimumab, but this isn't particularly effective, and it's actually very expensive. And therefore, there is a very high unmet medical need for specific and effective treatments for interferonopathies. And that's why we were attracted to this particular disease class. As I said at the outset, our targets are IKK-E or IKK-epsilon and TBK1. And these kinases play an important role in the regulation of the immune response. You'll all know that the immune response has evolved to protect us from infectious diseases caused by bacteria and viruses. And essentially the way it works is that on immune cells, on the surface, there are toll-like receptors, and in the cytosol of immune cells, there are nucleic acid receptors. And these detect the products arising from the infections by these organisms and use those, and when they receive those signals, they stimulate the immune response through signaling cascades, to activation of transcription factors, to the stimulation of gene synthesis, of genes, and then the synthesis of proteins that mediate the inflammatory response. In these signaling pathways, our targets are nodal enzymes. They're involved in both toll-like receptor signaling and nucleic acid receptor signaling. And so it seemed to us these were really good targets to inhibit in order to try and block this system in disease. In the disease, of course, unwanted pro-inflammatory signals turn on the pathways, and that leads to excessive stimulation of these pro-inflammatory gene syntheses. And they themselves are pro-inflammatory mediators, so we get into some kind of vicious circle where the system winds itself up. And our rationale was that if you could inhibit these enzymes, then gradually the system would turn itself down as the pro-inflammatory signals got weaker and weaker. So we wanted to tackle these targets, and we chose to use an FBDD approach. This was really because we wanted to firstly demonstrate the value of the approach, and secondly, because it had proved hard for other groups to find highly selective and bioavailable inhibitors using other hit ID methods such as high throughput screening. Now, I'm only going to talk about fragment-based drug design by implication. If you want to know the theory, I would commend this paper or review to you. But in essence, at DomainX, we start out by making the protein. Sometimes we have to use proprietary technologies to do that. We use that protein to develop assays based on biophysics or biochemistry, screen our fragment libraries, get structural biological information on how the fragments bind, and then use that to drive the drug design cycle. In this particular case, we screened the fragments using a high concentration biochemical assay against the IKK-epsilon enzyme. And we were also informed by a crystal structure which we had uniquely obtained. It's still the only known crystal structure of IKK-epsilon, which we used our domain approach to obtain. And then we optimized the compounds by molecular modeling and structure-based drug design using our cascade of enzyme and cellular assays. So this is the hit fragment I'm going to talk about today. I'm going to use this sort of top trumps representation so you can see some information about the compound. This is its potency against one of the targets as an illustration of the biochemical activity. This is the molecular weight. And these are two efficiency metrics that we use in order to quantify how good are we getting at finding a really nice compound. And this measure here, called ligand efficiency, tells us how well the compound binds to the target. And the value of 0.3 is considered very good. and the closer that we can get to 1, the better the compound is. We weren't able to crystallize this compound with the target, but we were able to dock it into the apoenzyme crystal structure that we had, and you can see that here. And you can see that it binds quite snugly into this groove. And I hope you'll be able to see that there are 2 opportunities to expand this compound, one in the northern part here, around this nitrogen, and the other one in this part here, which is around this position here. So we made analogues in both of these areas, starting with the northern part of the compound. You put a more bulky substituent here, and you can see you get a 10-fold improvement in potency, which is very exciting. And there's been no reduction in the efficiency of the compound bindings. That was a very nice start for us. We then said, well, what happens if we further substitute here? And we tried a range of things there. We tried directly adding aromatic rings. We tried adding substituents that would link through carbon or oxygen or nitrogen atoms. But the thing which really worked best for us was to add either an aryl or a heteroaryl ring with an amino linker. And that gave us a further 10-fold boost in potency. So we're now getting into the sub-micromolar range here. And this was very encouraging. However, we were slightly concerned by this drop-off in ligand efficiency. And one of the possible reasons for that is that, as you can see in this structure here, there is now a clash between these 2 hydrogen atoms in the enforced planar conformation of these 2 rings here. So we thought if we could remove one of these hydrogen atoms, then maybe we could get an improvement in potency. And what we found is if you replace this carbon here with a nitrogen to make this compound, you get another significant improvement in potency. And so with this scaffold here, you can see we've removed this clash, and we now have a 50 nanomolar compound. So far, so good. However, if you're familiar with kinase research, you might have think, oh, I've got a bit of deja vu here. I've seen this kind of scaffold before, this so-called aminopyrimidine scaffold, and you'd be right. And this is a really interesting example of what I think of as convergent evolution. So in a biologist, convergent evolution is when 2 organisms have evolved to be very similar as a result of adapting to a similar environment. So here's an example of a bat and a bird. They're actually biologically unrelated, but they both evolved to fly in quite a similar way as a result of wanting to capitalize on the benefits of being able to fly. In this particular case, our environment is an ATP binding site that's largely conserved in kinases. And although nobody else, to my knowledge, has ever started with this starting point, it's quite interesting that everything we tried to do to develop this compound failed to give us good compounds, except when we converted it into an aminopyrimidine scaffold. So I think what this is teaching us is that natural selection, if you like, takes us towards this kind of motif, which has previously been discovered by other people. For example, in the marketed drug imatinib, you can see there's a very common, very similar scaffold here. Nevertheless, our fragment-based approach has given us this cyano group, which is very distinctive, and that cyano group for this particular pair of kinases gives us exquisite potency, selectivity, and novelty, and actually, interestingly, relatively clear IP space. So had we not pursued the fragment approach, it's quite unlikely we would have found this particular substituent. Here's one of our better resolved crystal structures of a protein ligand complex, and I've angled this so you can see particularly well here, I hope, how the cyano group fits into this very, very narrow pocket in the kinase. And it's hard to conceive of any group other than a cyano group which could have done that, and also made a hydrogen bond to lysine 38, which sits at the back of this pocket. So by doing this, we've been able to get a very nice interaction with the target. So through this fragment approach, we've been able to find a motif that gives us high potency and selectivity. We now have to address the problems of getting good solubility, metabolic stability, and permeability. And one of the ways medicinal chemists use to do that is to lower the log D of a compound by introducing polar atoms and substituents. And what we did was to vary the molecule in a number of places, but the thing I'm going to zoom in on today in the time I have available is what we did with this phenyl ring here. So here's another view of that same crystal structure, and there are 3 possible isomeric pyridines that you can make, where the nitrogen goes here, here, or here. We made all 3 of them. Interestingly, this one, nitrogen here, is quite potent, but much less selective than the carbon analog. The one where you put the nitrogen here is much less potent, but very gratifyingly, the one where you put the nitrogen here is slightly less potent than the carbon analog, but it does crucially maintain that very high selectivity, and even more importantly, gave us very much improved metabolic stability. So looking at the in sort of stick drawing format, we've gone from a molecule like this to a molecule with this pyridine in place instead. And as the final part of the optimization, we looked at modifying the substituent in this position here. I won't tell you exactly what we put in the molecule to get to our candidate today, but by modification of this group, we were able to end up with DMXD011. You can see it's a potent inhibitor. You can see its log D has reduced enormously from the original potent compound, and the aqueous solubility is hugely improved. It's actually 3 orders of magnitude better than this compound here. And as a result of these changes, we've now got a very low clearance compound, a nice metabolically stable compound. And this was certainly suitable for us to now go ahead and do some proof of concept studies. Some of those were done in animal models, but the one I'm going to show you today was done in an ex vivo human assay. So it's obviously crucially important to find out whether this might work in patients. And the way we did that was by taking inflammatory cells, either from healthy people or from patients with these diseases, and then treating them with those cells with our compound and finding out whether we could block the immune mediator release. And here are those data. So there are different colors for different types of subjects. So there are healthy people, SLE patients, Sjögren's patients, and scleroderma patients shown here. And when you stimulate these cells, you get release of interferons, and you also get release of other mediators whose expression is controlled by interferon. So we get a very dramatic increase in the level of these inflammatory mediators. But when we apply the Domain X compound alongside that, you can see we almost completely blunt that response. So it's a very encouraging proof of concept. We got similar data when we looked at the interferon-stimulated gene profiles. So in summary, what we were able to do was to use fragment screening to find a very efficient and selective motif. We were able to use structure-based drug design to elaborate that to a potent series of inhibitors. And then by optimization of the PK profile, we were able to find a compound that could be used to demonstrate proof of concept in blood taken from patients. And I'd like to thank the Domain X team for this work and for you for your attention during the webinar. And I'm now going to field some questions. And I've got a couple of questions so far. The first was, does the Sano group engage in reversible covalent binding? And the answer is no, it doesn't. We've been able to demonstrate that by showing the reversibility by doing some experiments where we dilute the enzymes. Another question was, how important was LE and LLE metrics for narrowing down hits? And the answer was that ligand deficiency, the LE metric, is very important in our view. Lipophilic ligand deficiency is less important until you get into the optimization stage, and then we start to use it, but we don't use it to select the initial fragment hits because most of our fragments are of low lipophilicity. The last question was what FBDD screening method was used, and the answer to that is that we used a high concentration biochemical assay. However, we do also use biophysical methods for Domain X, and the one that we most favor is microscale thermophoresis because it is fast, uses very small amounts of protein, and gives us a very free choice of buffers to use. But we also use other techniques like surface plasmon resonance as well. And then the other question I've had is X-ray crystallography essential for FBDD? And the answer is no, but it helps. In this case, it was very helpful. We have done FBDD programs where we haven't had access to X-ray crystallography, and there it becomes a much more empirical process. And the fragment screening took us, I think, about 2 weeks. And I think my time is up. So I'm going to hand back over to James. Thank you for your attention.
Thanks. James, he's still on mute.
Good afternoon, everyone. Apologies for the slight delay there. I would like to thank the team at the MDC for inviting me to give this talk today and the opportunity to share my personal experience as part of this impressive webinar series. Before I begin, I would like to introduce Charnwood Molecular, which is a chemistry CRO based across 2 sites, one of which is in Loughborough, and the other, the larger site, is at BioCity in Nottingham. We have a total headcount of nearly 70 scientists, more than 85% of which have a PhD. Charnwood Molecular has been operating for 22 years, providing innovative expertise in both medicinal and synthetic chemistry, supporting client projects all the way from hit identification through to preclinical development and that first 200-gram batch of material for non-GMP toxicity studies. Today, I will be talking about synthetic chemistry aspects of drug discovery and early development projects based on my experiences. The discovery of a new drug is a long and challenging process, with most recent estimates suggesting that it can take somewhere in the region of 10 to 15 years at a cost of $1 billion to go from the identification and validation of a new target to an improved and marketed small molecule therapeutic agent. There are, of course, many steps in this process involving scientists working collaboratively across a variety of different disciplines and areas of expertise. And it is pleasing to see that almost all of these different steps have been covered in this excellent series of webinars. Now, what is notable about this graphic is the number of compounds that are reportedly synthesized at each stage of the process relative to the approval of a single small molecule drug based on current attrition rates in clinical trials. And when you think about the sheer size of this number, it is clear that chemistry is fundamental to the drug discovery and arguably the most important element in the whole process. When you consider the role of chemistry in this process, we can split the timeline in 2, based not only on the scale of synthesis that is required at the different stages in the continuum, but also on the very nature of the chemistry resource employed to execute these specific functions. And this gives you discovery chemists at the front end of the process, with the latter stages being covered by development chemists. Now, you may well say these 2 disciplines are one and the same, in that they are fundamentally synthetic organic chemists by training. And I would agree with that sentiment, conceding that there is some overlap, with synthetic chemistry at the heart of everything that they do. However, when we break down the specific skills required of the chemist to successfully navigate the earlier and latter stages of the drug discovery process, the differences between the 2 disciplines quickly become apparent. For example, the discovery chemist will complete the synthesis of many different compounds during the course of a single campaign using a toolbox of typically robust chemistries. Speed is of the essence to the medicinal chemist, with small libraries of compounds being designed, synthesized, and subsequently purified by automated chromatography. In most cases, the yield and cost of goods is largely insignificant, with the principal objective being to feed compounds into a screening cascade to establish a structure-activity or property relationship. An acute understanding of the underlying biology of the target is required to design and synthesize molecules with the correct balance of drug-like properties to reach the target of interest and elicit the desired therapeutic effect. Computational tools are also applied routinely at this stage to model the binding of compounds to the desired target, just as Trevor's shown you, generating new ideas for synthesis to identify highly selective hand-in-glove interactions. Lastly, design and synthesis must always be conducted with an understanding of the potential for DMPK liabilities, such as rapid clearance, cytochrome P450 inhibition, or toxicity issues, including on and off target effects. In contrast, the development chemists will focus specifically on the synthesis of a small number of compounds that have been nominated as candidates at the end of a lead optimization campaign. Yield and the cost of goods are everything to the development chemist as they strive to design robust, highly efficient, and cost-effective routes to the candidate compounds. The development chemist therefore needs to develop an intimate mechanistic understanding of each stage in this synthetic scheme, as well as being able to identify the impact of each processing parameter on the outcome of the reaction. And they will typically run a series of range-finding experiments using design of experiment principles to identify the optimum conditions for each step. All of this requires a good understanding of analytical chemistry and structural elucidation to identify any impurities that may be formed during the course of the reaction and to design them out of the process. Lastly, as the heavily optimized process moves towards clinical trial and GMP manufacture, development chemists need a good understanding of the regulatory framework as well as basic knowledge of chemical engineering to better facilitate the transfer of their process to the pilot plant and beyond. So what are the differences do we need to consider between discovery and development chemistry? It is clear that the transition from discovery to development occurs with a significant increase in the amount of material required and the scale at which the chemistry must be conducted. But what does this mean? Well, for the discovery chemist, you most likely conduct your chemistry in kit similar to that depicted below, typically making around 20 mg of each final compound to support all of the necessary screening activities. You may therefore assume that preparing larger amounts may simply require a larger version of the same kit, and this assumption would look something like this cartoon. Pleasingly, however, reality paints an altogether different and much safer picture, with large-scale chemistry typically conducted in jacketed reactor systems similar to the one shown here, which is what we have at Charnwood Molecular to support our client's project. And as you move through development, these vessels get bigger and bigger, and we've all got that one colleague who takes the concept of social distancing to the next level. Okay. So let's go back to the beginning of the drug discovery process and think about where the synthetic chemistry journey begins. 2 years ago, Brown and Bostrom looked at the origins of small molecule clinical candidates from 2016 to 2017. And what they found is that most clinical candidates come from existing starting points, including fast follower or Me Too approaches. But what if you want a fresh starting point against a novel target? In that case, you might want to look at one of several different screening platforms, such as high throughput screening, fragment-based screening, such as what Trevor's just described, or the use of a DNA encoded library, for example. And if you think about the chemical space that the compounds resulting from these screens might occupy in terms of, say, for example, their physical chemical properties and molecular weight, you will end up with something like the following distribution. And what we see is the boundary for the rule of 5 space for orally active small molecules, which was formulated by Lipinski in 1997. Within this space, we see clusters that represent traditional high-throughput screening decks, including focus libraries. And if we continue downwards and to the left, we enter the fragment space, where the project will follow a similar trajectory to that described previously. At the end of the other end of the spectrum, going out beyond the rule of 5 boundary, we move into an area of space typical of some natural products and what you might find as the output from a DNA encoded library, an area which, despite the low numbers of candidates reported in 2016 to 17, is growing, in my experience, in popularity. Although the nature of your target may determine the type of screening platform that you use, and indeed the chemical space into which you move, the synthetic chemistry underpinning all discovery projects, regardless of the direction in which they travel, is largely the same. Now, what do I mean by this? Well, papers published in 2011 and 2016 by Ruffley and Brown, respectively, revealed there are essentially 4 to 6 reactions that discovery chemists turn to time and time again to support the progression of their projects towards development. And these reactions are highlighted here. Notably, the more recent of these 2 publications revealed that the discovery landscape was dominated by the amide bond-forming reaction in 2014. And this is a trend that continues today, with the humble amidation reaction appearing in almost 1/3 of small molecule therapeutics approved so far this year by the FDA. Now, because of the importance of the amide bond to drug discovery, it has become a regular feature of technical interviews where the interviewee is often asked through a grimace smile to tell the interviewer their favorite way to make an amide. And it is true that this transformation can be facilitated by a multitude of different coupling reagents, mainly from the corresponding carboxylic acid and amine. However, a deeper consideration of this transformation serves as a good example of the fundamental difference that often exists between the approaches taken by discovery and development chemists towards their work, a difference that is best illustrated using a tool known as a transformation map. When I worked at Pfizer and Cancer Research UK on discovery projects, I would often find myself in possession of a small pot of an advanced intermediate bearing a carboxylic acid, designing a small library of final compounds for SAR analysis via an amidation reaction. On paper, this reaction is always considered quite simply as it is shown here on the slide. A plus B goes to C. And this is largely fit for purpose in the discovery setting. But this simple interpretation belies a complex series of mechanistic events underpinning the transformation, the importance of which we will come to shortly. Nevertheless, in the discovery setting, you run this reaction in a Radley's carousel with your favorite coupling agent and a bunch of amines selected by design to interrogate a specific parameter or address a known liability. After stirring overnight, you take a UPLCMS of your reaction and you get the following chromatogram. And you look through the peak table for the corresponding mass spectrum and you see the mass of the desired product and you're happy. You might not think too much about the origins of the other peaks in the trace because you have automated chromatography, which means that these issues quickly melt away to afford a beautiful chromatogram of the desired product ready for the screening cascade. But what if this compound is the one? What if this compound is the clinical candidate? You and your colleagues in discovery get really excited. You prepare a technical package and you email it over to your development partner asking for 200 grams of this material. The email arrives, and whilst they naturally share your enthusiasm, they have many questions. Now, in contrast to discovery, the transition to development requires a detailed understanding of the rate processes driving the formation of the unwanted side products, with the specific aim of favoring the desired transformation over all of the other reactions, thereby optimizing the yield, simplifying the purification process, and ideally eliminating the need for column chromatography. And this understanding can be platformed on a transformation map like the one shown here, where the green processes represent the desired transformations, amber represents semi-desirable pathways that have the potential to undermine yield, and the red pathways are completely deleterious. In this example, the formation of the NA-sal urea is unwanted and can be attributed to the use of a carbodiamide coupling agent. The solvent also plays a part, with solvents with a high dielectric constant favoring the rate process K4. The use of a weakly nucleophilic amine would also further contribute to this issue, allowing K4 to dominate over K1, leading to the issue that you see here. Of course, switching the solvent or the coupling reagent or both could therefore improve this situation, and this is reminiscent of many problems that I have seen in development, where my team would devise a solution by running a series of range-finding experiments in parallel as part of a systematic design of experiment process with the aim of favoring K1, the desired reaction. But maybe this is an issue that should be addressed during the discovery phase, building a better understanding of the transformation and using that understanding to semi-validate a development-ready process, saving considerable time and money regardless of what stage you're working at in the drug discovery process. And this is something that we did recently at Charnwood Molecular on a project. Now, in this particular case, the project required the design and synthesis of a compound series based on a tri-substituted phenyl ring. And the retrosynthetic analysis of this is shown in the scheme, taking us back to this fluoronitrobenzene bearing an electron withdrawing group. In the forward sense, this compound was subjected to sequential hydrogenation, amidation, and SNAR reactions, allowing for the diversification of the highlighted position at the final stage of the sequence to introduce a variety of different heterocycles. Now, the existing technical package revealed that the yield for this amidation stage was very low, regardless of the incoming carboxylic acid. And this was something that was generally accepted by the original discovery team, who were happy to purify the reaction mixture by chromatography at the expense of the yield. When we considered the transformation map for this reaction, as well as some anecdotal information from familiarization experiments conducted in our own lab, which indicated that the active ester was formed quantitatively, it became clear that the poor reactivity of the incoming aniline was responsible for the low yield, with K2 being with K2 being really low. This is perhaps not surprising when you consider that the aniline is flanked by two electron withdrawing groups, severely attenuating its nucleophilicity. So now that we understand this issue, we are in a position to devise a solution, and we envisage two possibilities. One, we could enhance the nucleophilicity of the incoming aniline, or two, we could increase the electrophilicity of the carbon or carbon of the acid. And this thinking led us to the award-winning amidation reaction developed by Buttner and his team at BMS in New Brunswick. In this well-written letter, Buttner describes the elegant use of TCFH and NMI to allow for the in situ generation of a highly reactive acyl imidazoline, which readily reacts with non-nucleophilic amines to give the corresponding amides under neutral conditions. And the application of this method to our weakly nucleophilic fluoroaniline proceeded well, with the complete consumption of both starting materials. However, notably, we did observe the appearance of a pronounced impurity indicated here in the chromatogram. Now, it would have been tempting at this stage to simply purify this material by column chromatography, taking the resulting clean material forward to build a small library of analogues using the nucleophilic aromatic substitution reaction. But if we did this, we would have sacrificed almost half of the potential yield, undermining the efficiency of the reaction. So what we did instead was consider the transformation map in more detail, and something in the transformation map that would otherwise not appear in the generic scheme, highlighting the importance of the transformation map, suddenly captured our attention. And this was the N-methyl imidazole. Now, NMI is a highly Lewis basic amine with a pKa of 7.2, allowing it to act as a nucleophile and undergo an SNAR with the intended aryl fluoride product. And with that, we had identified the impurity. But what was the benefit, you may well ask? Well, as I'm sure you're aware, the first step in an SNAR reaction is the rate determining step. And this is why an aryl fluoride performs effectively in an SAR, more so than the corresponding aryl chloride, bromide, or iodide in that order. And this is the reason, is because of the polarization of the aromatic carbon, making it more attractive to the incoming nucleophile. So in short, we envisaged that this apparent imidazolium impurity would behave in the same way as the desired aryl fluoride, and to cut a long story short, it did, with both compounds undergoing the desired SNAR reaction to give the corresponding products required as part of the library build for the original scheme. Furthermore, this was done without the need for any purification whatsoever, so we were able to telescope this process without any column chromatography, and it was subsequently scaled up to 100 grams without issues to support the development phase, highlighting the benefit of running these transformation maps to develop an intimate understanding of the work that you're doing. So hopefully I've convinced you that transformation maps of this type are incredibly useful and can be applied to any reaction at any stage in the drug discovery process, providing an intimate understanding of the different pathways available to the many components of the reaction above and beyond the desired transformation. Understanding and minimizing the rate factors driving the different diversionary pathways ultimately not only facilitates the optimization of the transformation, but also represents, in my opinion, good practice. And this is something that I've always done myself as a chemist, and that I actively encourage all of the chemists working at Charnwood to do too. And as you can see from this last example, this way of thinking can also present opportunities, allowing chemists to build more robust and development-ready processes. Okay, so in conclusion, the synthetic considerations of discovery and development chemists are different, and this is largely driven by the different pressures faced by chemists at their respective stages of the drug discovery process. Regardless of these differences, synthetic chemists and synthetic chemistry should always be designed and executed with the aim of developing robust and highly efficient processes. And a lot of this can be conducted upfront, essentially as a paper exercise using the existing literature with the potential to deliver considerable savings in time and money. Hopefully, I've convinced you that transformation maps provide a detailed understanding of the potential challenges and opportunities in any reaction. And next time you run a reaction, I would encourage you to build a transformation map and think about the rate processes driving the different reactions that could potentially take place in your flask. Lastly, I would like to encourage you all to always engage your colleagues in the wider synthetic community and work collaboratively to design chemical processes that work as efficiently and cost effectively as possible, regardless of the stage that you may be working at in the drug discovery process. Thank you for listening.
Okay, thanks, James. Let's just take a question.
Okay, so I've got a couple of questions here. What are the most common showstoppers for scaling up a process which needed to be developed out? I've never seen full showstoppers in that there's always a solution to a problem. One of the things I love about process chemistry, in a sense, is that if you understand it well enough and you develop that intimate understanding, you can more or less predict how applying a single pressure to a reaction, what it will do in terms of the trajectory that the reaction takes. So developing that understanding is fundamental. And with that in mind, once you understand the problems that you have and what's driving those problems, you can design them out of the process and move things forward. Another question is, do you consider the synthesis of amides with the use of carboxylic acid derivatives to be disadvantageous on a larger scale instead of acid directly? I'm not entirely sure what that means. The use of, I've conducted many amidation reactions on the large scale in GMP manufacture, and these are reactions that I've conducted up to 100, 200 kilograms in previous employments. These have always been done more often than not with the simplest possible and most atom economical coupling reagents that you can get. So it might be that you convert it to an acid chloride, which is perhaps what you mean, and then react that with an amine, and that does work well. One of the things to bear in mind in that case, and something that I've seen in the past, certainly for GMP manufacture, when you're using acidic media with, say, THF or 2-methyl THF as a solvent, this can lead to the ring opening of THF to form alkyl halides, which are genotoxic impurities. And in another program that I ran, we ran an amidation in acetonitrile, and this led to the formation of acetamide, which is also a genotoxic impurity. So you need to be aware of the liabilities associated with the different coupling agents that you use.
Okay, James, that's great. Thanks very much.
Okay, cheers.
Okay, thank you.
Okay, good afternoon, everyone. Thanks for joining us, and thank you to the organisers for the invite. So following on from the previous two speakers, I'm now going to talk about formulation design for preclinical and early stage clinical studies. So Key Pharma, who are we? We are a contract development and clinical manufacturing organisation. We're a family-owned business based in the UK, just outside of Chester. We were founded in 2002 by Mike Rubinstein, who's the Professor of Pharmaceutics and Chemistry at the Liverpool John Moores University. We've now got over 180 people working on site, and we're experts in formulation of molecules for systemic and localized delivery. We consider ourselves as specialist formulators of problem molecules, which include small molecules, live biotherapeutics, antibodies, and other large molecules. And we can perform GMP clinical manufacture of all the products that we develop. So I've tried to put into context a bit here where we sit in the development pathway. So API synthesis and characterization would normally be performed at another CMO, such as those we've already heard about this afternoon. So Key would receive the API and will formulate that for preclinical studies, aiming to maximize dose and absorption in the animal models. The preclinical formulation or the method of manufacture we would then transfer onto a recommended CRO for the DMPK and tox evaluation. Results from that DMPK and tox study will then help to guide the clinical formulation development, and we'd normally start that with the pre-formulation studies. These will include things like powder characterization, excipient compatibility, and also solubility studies, but quite often the solubility studies would have been performed as part of the preclinical development. Once we've generated some data from the pre-formulation studies, we then move into the formulation development, and the pre-formulation study results will help guide us into the formulation development. We optimize the formulation approach, and we generate some early prototypes. We then moved into the feasibility batch, which will be scaling up the lead compounds at a smaller batch than a GMP process, but it would mimic the actual GMP process itself. Those samples would then go onto ICH stability prior to the actual GMP manufacture. So moving on to preclinical formulation, we have some general considerations. So for a preclinical formulation, we want to actually maximize the dose and absorption. Typically, a liquid dose will be required because it will be dosed via an oral gavage. We're often dealing with a poorly water-soluble drug with typically very little API actually available. And we want to create a formulation with minimal time and efforts, as it's a proof of concept formulation, but we still need for it to achieve good absorption. We also need to pay careful consideration to the excipients that we use due to potential toxic effects in the different species, which can be different to what you'd see in humans. So some useful data to know. Things like chemical structure. Does the molecule contain a chromophore for analytical purposes? Solubility data, if there's some that already exists. So the aqueous solubility, does it have a pH solubility profile? Any solvent data, solubility data in different solvents? The log P, pKa and melting point. Is there any CaCO2 data? How much API do we have to play with? What's the maximum target dose required in the animal species? And actually, what is the intended preclinical species as well? So Key have a few platforms that we use for preclinical formulation. The main one being solubility screening. This is obviously the simplest option, and where we would start, we include a range of different solvents and surfactant solutions, lipids as well, and we're trying there to maximize the absorption of the poorly soluble drug candidate. We can also include precipitation tests at this point, depending on requirements for the formulations, particularly if it's dosed orally. We then have the solid dispersion and nanoparticle formulations. Now these are more of a fallback option if the solution or lipid formulation isn't viable. Typically a more complex route, which we're aiming at poorly soluble BCS class 2 molecules. However, those formulations can help us to bridge towards a phase 1 formulation. The platforms are bespoke. The excipient selection is very much based on the API characteristics, the animal species, and the downstream requirements. We use minimal API and we turn around the work very rapidly. So an example here of a screen that we carried out, solubility screening. So here we've compared what we call a macro method, which is a traditional equilibrium solubility study, with analysis either by UV, which you can see in the yellow bars, to HPLC, which is in the darker blue bars. And then we compared that to our screening method, which is the pale blue bars. So in the screening method, we're actually targeting a maximum of 20 mg per ml. So if we achieve full solubility, that's the maximum solubility we will achieve. But we do see the same trends for both methods, both analytical techniques across all the samples. With the advantage being, if you can look in the table on the right there, is that we're using at least half the amount of API for the screening method, and we're only taking about a day and a half to do that work. So now if we turn to early clinical studies, some general considerations here, which are very similar to those for the preclinical studies. Things like the patient health benefits and requirements. What is the actual goal of the phase 1 study? Is it a proof of concept only? Is it to bridge to a phase 2 study? Do we need to have as close to a final formulation as possible? What is a preferred route of administration? What's the target product profile? And this is an essential piece of information and some indications of what's involved in that, requirements of that on the right there. So things like indication, patient needs or limitations, for instance, if the patient suffers from dysphagia, the dose range and frequency, the patient population, target release profile and target absorption site. The balance between the chance of success against the degree of acceptable risk, and the balance of the cost and time to get into that phase 1 study. They're all very much linked. The consideration is the submission strategy, commercial considerations, and API availability. And finally, the regulatory timeframe. So some useful data to know before we go into some formulation is the physicochemical characteristics of the API. You need to marry the available data with the desires of the target product profile. And the rest of the data really is very much similar to what we want for preclinical studies. But additionally, things like the particle size, distribution, shape or density, flow characteristics of the API, particularly if it's going to be an oral solid dose, the polymorphic form, and any stability data if force degradation studies have already been performed on the API. Biological characteristics, so things like permeability, CACO2 cell data, any potential efflux mechanisms, and obviously the existing pharmacokinetic data which we should have following the animal studies. So the development path of the formulation will very much depend on the API classification. So this is probably not going to be new data for you, but it's looking at the developability classification system. And the data obtained from the preclinical solubility study can help to define the DCS classification together with the target dose from the TPP. So if it's a class 1 molecule, that's great, everybody's favourite, and hopefully shouldn't be too challenging to develop a formulation for that. Class 2, majority of molecules will fall into this class, can be a bit of a challenge, but equally quite interesting to work with. For class 3 molecules, we don't generally see those too often, and permeability can be an issue. And if it's a class 4, unfortunately, you're very unlucky if it's a class 4. There are ones that exist out there, but they're notoriously quite difficult to formulate. So looking at class 1, basically the world's your oyster. So if you want to get into phase 1 clinic quickly, API in bottle, API in capsule, really quick formulation to get through to a phase 1 study as quickly as possible. And as we move down that slide, the formulations get more and more complicated, but you actually might want something that's a modified release system. Obviously, as we go down that slide, we're getting increasing duration, cost, complexity, and risk with the formulation. For class 2 compounds, majority of new candidates do fall into this category. The absorption rate will drive the selection of approach. So that depends on whether it's dissolution rate limited or solubility limited absorption. Depends on whether we're looking at a particle size reduction or lipid solid dispersions or a microenvironmental pH control. Clearly, the more complex formulations will have a higher risk associated with them, but the rationale for choosing which route we go down must be based on the available data, and it can be quite a complex decision tree. We review each API on a case-by-case basis, and the rapid screening approach in the preclinical study is available and can help with selection of some formulations for the class 2 molecules. So for class 3 and class 4 molecules, there's an additional consideration in that has the actual correct lead formulation been, correct lead compound been selected. So could the solubility be improved by salt formation or by co-crystals, for instance? And could the permeability be improved by delivery as a prodrug? If we look at class 3 compounds, things that consider are things such as can permeation enhancers be incorporated, or even can the transit time be reduced to increase the absorption potential? For class 4 compounds, can combinations of permeation enhancers and solubility methods be employed? So I mentioned earlier about the importance of balancing the risk, time and cost. So if we're looking at the risk, if a more advanced formulation is prepared for a phase 1 study, then there is an easier transition to phases 2 and 3. However, if a simple formulation is used for phase 1, then for later stages, it may actually be necessary to move to a more complex formulation. And by doing this, it actually may lead to a difference in the bioavailability in man. We also need to be aware that as you go from phase 1 through to phase 3, we're scaling up, and we'll typically be moving from manual to automated processes. In terms of cost, if we start with a simple formulation for phase 1, the formulation costs for phase 2 will be greater because more complex formulation typically below the cost for phase 2. It also depends on the company itself, so often getting through different phases will release milestone payments, but there's also costs as IP considerations if there's an IP portfolio. And in terms of time, so the speed to clinic can be key if we're needing a milestone payment. Enabling technologies are typically more complex and will require longer to develop. And also availability of the technology might be key, and equally API supply. So in summary, gone through some critical factors for selecting the appropriate formulation, whether it be for preclinical or clinical studies. And these are things like the patient needs and safety, dosage stability to be delivered, the target release profile, the complexity of supporting analytical or screening methodologies, the overall development strategy and timelines, readiness to assume risks, and financial implications. And thank you for listening. And just check to see if there's any questions. So there's one question here. Can you touch upon some differences in formulation issues for IV administration versus oral? So in terms of IV, clearly, apart from the formulation requiring to be sterile, there's a very much an excipient selection is quite key for IV formulations. There's limited excipients that are acceptable for IV administration compared to what can be taken orally. So that's really got to be narrowed down for that. Also, it requires the compound to be soluble. So if it is a poorly soluble molecule, you are going to need to incorporate some sort of IV acceptable solvents in the formulation and also get it to dissolve and to be administered. There's also things you can do. You also need the API to be stable in solution, but that's where things like lyophilization come into play, because you can have a compound that's lyophilized and reconstituted immediately prior to administration if there is stability issues in solution. Is API quality important in early clinical studies? That's also another question. Ideally, yes, it's beneficial if it is pure, as pure as it can be, because sometimes if there are impurities present, that can actually change the solubility of the compound. So it might change how the molecule will behave later on when you're formulating it, when you're getting towards GMP manufacture. If you get a pure compound versus an impure, you can get different polymorphs as well, depending on impurities maybe in the material. So although you kind of think early stage, or it doesn't really matter, it makes things a lot simpler later on downstream if it is as pure as possible. Because I can think of one case where we worked on a project and the purity did change, and it did have an effect on sort of dissolution rate from the formulation.
Okay, thanks, Alison. Thanks very much for that.
Okay.
Sorry, so I'll show my slide. Thanks to the speakers today, to Trevor, James and Alison, for steering us through this week's MDC Connects webinar. Next time we'll be focusing on understanding the PK-PD relationship. We've got 3 talks. Principles and Modelling of PK and PD Relationships from Graham Trevitt at Xelogesis. Jenny Worthington from Axis Bio is going to help us understand PK-PD using preclinical models, lessons for efficacy studies. And finally, Neil Gingles from here at Medicines Discovery Catapult, who will be taking us through the evaluation of clinical and preclinical PD endpoints using non-invasive imaging. So thank you all once again for listening and to the speakers, and see you next week.